For the crosslinking followed by immunoprecipitation procedure, cells at 80 to 90% density are incubated with 1 millimolar ice-cold DSP in PBS calcium-magnesium or DMSO-only vehicle control. DSP is a homobifunctional, membrane-permeable, and thiol-reducible crosslinker that crosslinks free amines within 12 angstroms of each other. Following crosslinking, cells are lysed, and immunoprecipitations are performed using standard controls. Protein complexes are then resolved by reducing SDS page. Reducing agents cleave DSP, allowing for the identification of individual proteins by immunoblot.
Hi, I'm Stephanie Zlatic from the laboratory of Dr. Victor Faundez in the Department of Cell Biology at Emory University.
And I'm Pearl Ryder, also from the Faundez lab.
Today, we'll be showing you a procedure for the isolation of multi-protein complexes using controlled crosslinking followed by immuno-magnetic affinity chromatography.
We use this procedure in our laboratory to study transient labile protein interactions. So, let's get started.
Begin by labeling the bottom of the cell plates to indicate the cell type and specific experimental condition. A positive symbol here indicates DSP treated. Include a negative DMSO-only vehicle control. Add media to the plates to prepare them for cell seeding. Plate HEK cells seeded at a 1:5 dilution to allow isolation of 500 micrograms of protein per standard tube reaction. A single tube may suffice to identify the putative binding partners of a protein of interest by immunoblot. For mass spectrometry analysis, increase the number of standard reactions at least 10 times. Also prepare stocks of most of the solutions used in the crosslinking experiment. These include PBS supplemented with 0.1 millimolar calcium chloride and 1